Hello students. So welcome back to the channel. In this video, we are going to study about C4 and camp plants. Now see, ever since plants, they have first moved to the land about 475 million years ago, they were trying to adapt themselves to the problem of the terrestrial life, means dehydration problem. They were trying to do the adaptation. They are showing many anatomical and metabolic adaptation. We have studied in the previous video, C4 plants have developed a special type of anatomy, that is Crane's anatomy, to show the adaptation in the problem for the dehydration, to do the compensation. An important example is the compromise between the photosynthesis and prevention of the excess water loss. Carbon dioxide required is entering through stomatas. And even the stomatas are the site from where the transpiration occurs, means loss of water so that the ascent of sap can continue. On a hot and dry days, most plants will close their stomata, and this is a response to save the water. This response also reduces carbon dioxide availability for the photosynthesis. So with the stomata even partially closed, carbon dioxide concentration start decreasing in the airspace within the leaf and oxygen concentration increase. So what happened in this condition, what they are doing some wasteful process, that is photorespiration. Why we say it is a wasteful process? Because neither ATP is synthesized, neither sugar is synthesized. So they have developed a special type of anatomy and special type of plant to cope up the hot and dry climate. So C4 plants are the one which are developed. In some plants which are found in hot and dry climates, we are finding they are showing alternate mode of carbon fixation so as to minimize photorespiration and to optimize the Kelvin cycle. They are called C4 plant. Why? Because the first fixation product is a 4-carbon, that is oxaloacetic acid. So when the weather is hot and dry, C4 plants will keep their stomata mostly closed to save water, but they continue to prepare sugar because they have developed two types of the chloroplast. Dimorphic chloroplast they have developed. Mesophyll cell chloroplast is, here the chloroplast, mesophyll cell chloroplast is with grana and bundle sheet cell chloroplast is without grana. So they have developed dimorphic chloroplast. An enzyme in the mesophyll cell, that is pepcase, is having very high affinity for carbon dioxide. So here what is going on? This is the enzyme pepcase, which is having very high affinity for carbon dioxide, will combine with 3-carbon compound, that is phosphoenol pyruvate, to form oxaloacetic acid, which is then changed to malic acid or aspartic acid and is transported to the bundle sheet cell. Now, in bundle sheet cell, decarboxylation will be done. The carbon dioxide enter into the Kelvin cycle, and 3-carbon, that is pyruvate, is then returned to the mesophyll cell where it is regenerated again back into the PEP. And Kelvin cycle is the method by which they are preparing the sugar. So thus the carbon dioxide concentration will be remaining always high in the bundle sheet cell, and they are avoiding photorespiration. Another type of plant which is able to survive in hot and arid condition, these are succulent plants, and they are generally belonging to the family Crassulacean family. Okay, so these are also known as chem plants. Chem plants means Crassulaceans acid metabolism. So they are generally seen in the Crassulacean family, and they are doing what? They are actually having scotoactive stomata. What do you mean by scotoactive stomata? Means those stomatas which remain open during the night and closed during the day. So how do they survive? What they are doing it in the night time, see the diagram, in the night time, the stomatas are open, the carbon dioxide is accepted by phosphoenol pyruvate and is becoming into oxaloacetic acid, that is 4-carbon compound, which is changed into the malic acid, and malic acid accumulate into the vacuole. In the daytime, when the stomatas are closed, then what happens? This malic acid will undergo for decarboxylation, and carbon dioxide will enter into the Kelvin cycle. Okay, and 3 carbon compound will be regenerated again to make a PEP. So in this, you are able to see in pineapples, you can see in many cactus and other succulent plants. Succulent plants means those plants which are having a strategy to store water, and they are adapted to very dry climates. Now, if you see, both are using the same process, but how do they differ? Why we are calling two different type of plants? In C4 plants, if you see in C4 plants, we are using two cell, mesophyll cell and bundle sheet cell. In mesophyll cell, acid formation is done, that is oxaloacetic acid, which is given to the bundle sheet where the Calvin cycle will operate. But in chem plant, the process is occurring in one cell only, that is mesophyll cell. It is only a time difference. In the night time, they are making the organic acid, and in the daytime, they are doing the Calvin cycle when the stomatas are closed. So C4 plants will show separation in the space. They are showing separation in the space by using two different cell but same time. Chem plants are using temporal separation, means they are using two different time but cell is one. So it is occurring in the same cell and is occurring in different type of cell. So I hope so. Whatever be the methods, at the end, Calvin cycle is the only one by which glucose will be produced. So I hope up till here your topic might be easy for you and you have understood alternate methods which is developed by the plant to survive in hot and dry climate is making C4 pathway and chem plants. I hope so this video might be helpful for you. Thanks for watching.